Welcome back to the Remdesivir files. Today we are diving into the analytical methods used to test Remdesivir, how scientists ensure its purity, potency, and consistency. To shed light on this process, we have Saakshi Sharma, an expert in pharmaceutical analysis. Welcome, Saakshi. Let's start with the basics. What are some of the key analytical techniques used to assess Remdesivir's quality, and why are these methods so important?
Thank you for having me. When we talk about ensuring the quality of a drug like Remdesivir, we are really talking about two key aspects, purity and potency. These are critical to both safety and efficacy. One of the primary methods used in testing Remdesivir is the high performance liquid chromatography, or what we call HPLC. This technique is widely used to separate, identify, and quantify components in the drug sample. HPLC allows us to measure the concentration of the active ingredient, remdesivir in this case, precisely and identify if any impurities that might be present. This is crucial because even minor impurities can affect the drug's performance or, in rare cases, pose a safety risk. Another method that plays an important role is mass spectrometry, or what we call MS. Mass spectrometry is typically used in conjunction with HPLC, forming a tandem technique called LC-MS. This combination helps identify the molecular structure of compounds, ensuring that remdesivir not only has the correct active ingredient, but is free from unwanted byproducts or contaminants. Mass spectrometry can also be used to detect any degradation products that might arise from storage or manufacturing processes which could affect the stability of the drug.
Fascinating. It sounds like these methods help ensure that remdesivir's formulation is safe and effective. But what about batch consistency? How do scientists ensure that every batch of remdesivir remains the same, even if it's produced at different times or in different facilities?
Batch consistency is absolutely crucial in pharmaceutical manufacturing. This is where stability testing comes into play. In the case of remdesivir, stability studies are conducted to understand how the drug behaves over time under different environmental conditions, like varying temperatures, humidity, or light exposure. Analytical techniques such as HPLC and UV visible spectrophotometry are used to monitor the stability of the active ingredient. Regular testing for consistency ensures that the potency and purity remains within the acceptable range for each batch, even if it's produced months apart. In addition to this, the pharmacokinetic profiling of the drug is routinely analyzed through these methods. Understanding how the drug is absorbed, distributed, metabolized, and excreted from the body ensures that every batch behaves similarly in the patient's system. This allows clinicians to be confident that each dose of remdesivir has the same therapeutic effect regardless of when or where it was manufactured.
Interesting. So, it's not just about making sure remdesivir works as expected in individual patients, but also about ensuring that each batch is identical in terms of quality. We also speak a bit about how these tests are regulated and who ensures the reliability of these analytical tests.
That's a great question. The analytical testing of drugs like remdesivir is highly regulated by health authorities such as the FDA in the US or the EMA in the Europe. These agencies set strict guidelines for testing protocols to ensure that every batch meets safety, efficacy, and quality standards. The tests themselves are often carried out by both the manufacturer and the independent lab. Additionally, the good manufacturing practice, or what we call as GMP guidelines, require that drug manufacturers maintain detailed records of all quantity control tests which are performed, including the analytical testing as well. Furthermore, testing methods and results are subjected to audits by regulatory agencies to verify compliance with these standards. So it's not just about the lab performing the tests. There's a rigorous framework in place to ensure transparency and accuracy as well in these processes. The reliability of the results is a paramount, because any deviation can lead to significant regulatory scrutiny or, more critically, affect patient safety.
Thank you for the detailed explanation. It's clear that the analysis of remdesivir involves a complex series of steps, ensuring that it remains effective and safe across different batches. Any final thoughts on how these techniques contribute to the overall success of the drug, especially in a pandemic context?
Absolutely. In a situation like the COVID-19 pandemic, where treatments are in high demand and fast tracked for approval, maintaining rigorous quality control through these analytical methods is crucial. They not only ensure that remdesivir perform consistently as expected, but also provide reassurance to healthcare professionals and patients. With the right analytical techniques in place, we can trust that each dose delivered is both safe and effective, which is crucial when millions of people are relying on these treatments to recover.
Thank you so much for joining us today and for your deep insights into the analytical methods behind remdesivir. That's all for today's episode of the Remdesivir Files. Stay tuned for more deep dives into our next episode.